And so next up, we have Holly Shugart, who is an assistant research professor from Penn State. And Holly will be talking to us now about host plant interaction and emerging risks. Holly, are you ready to share your screen?
I'm ready, yes.
All right, go for it.
Okay. Good morning, everyone. I'm excited to talk to you today about research I've been doing on host suitability and survival of spotted lanternfly on selected specialty crops. We're going to talk some about adults and some about nymphs. What we're not going to talk about is host preference. We didn't give any of these insects choices about which hosts they were on. We bagged or caged them to hosts and tested their performance. So as Julie introduced, this insect is highly prolificous, and being invasive, that combination of factors makes them really hard to predict what they're going to do and how they're going to behave. So we found that adult survival is variable in different hosts. Previous studies have demonstrated this. We're trying to get into some of the details. And we worry that this variability will make establishment to new regions and habitat complicated. And that establishment will be linked to host availability and suitability. They can survive on a wide variety of agricultural, forest, and green industry hosts. We've seen that in all of the places they've impacted so far. How long they survive will mediate specific impacts. So the longer they survive, the more they have a chance to damage the host. And the longer they're alive on a host can drive expansion. If they don't survive well, it can slow expansion to new regions. So nymphal survival is also important. We can also look into development. So remember, these insects overwinter as eggs, and they hatch out as nymphs, and they have 4 nymphal instars, and then they become adults. And so as nymphs, they don't have wings, and they can't reproduce, right? So which hosts can the nymphs survive on? And survival, of course, is going to risk establishment into a new habitat. Think of if an egg mass arrives in a new location, what happens when the egg mass starts hatching out? Can the insects survive in that habitat? Will they find something to feed on? And importantly, can they survive to adult? Because once they're an adult, they can fly and they can reproduce, and that changes the possible impacts if they can survive to adult. So here's an outline of my experiments. We're going to talk about adult survival. So first, we're going to look at adult survival tracked on 9 hosts. And then this until all individuals are dead, that tracks for all of the experiments we're talking about today, okay? And then we were curious about our citrus results, and we repeated it. You know, you're supposed to be able to repeat your experiments and in science. And sometimes they're the same, and sometimes they're different. We'll talk about that. For the nymphal work, we looked at survival and host impacts by caging 50 first instars and then tracking them throughout their nymphal time and how long they survived. And if they survived long enough, we also looked at their development and tracked those developmental milestones to see how that went. So for adults, we looked at 9 different hosts, and 3 of them were grapevine. So 2 wine grapes, Sauvignon Blanc and Chardonnay, and we looked at muscadine, variety Black Beauty. We looked at fuzzy kiwi, we looked at hops, and here we looked at Cascade and Centennial varieties. We looked at citrus, and this is sweet orange, variety Valencia. We looked at avocado, variety Haas, and almond, variety Mission. So these are our 9 hosts that we'll compare survival on for adults. And then remember, we also have adult survival on citrus, and we did this repeat. And part of why we did this is because the trees were older and they were fruit bearing. So we were worried about impacts like fruit drop, for example. We also, in this test, gave the insects full access to the trees rather than bagging them to a section of the tree to see what the impact there might be. Okay, and then we looked at nymphal survival and development. We only looked at citrus and avocado for this test. Okay, so now let's talk about the data for adult survival. So this is a Kaplan-Meier survival plot. You're going to see a lot of these today. So you're going to see two main types of charts, and this is one of them. And what this shows is the survival rate and the days survived, and it allows us to visualize their survival over time, right? So did they have a gradual decrease or a very steep decrease? And then how long did they survive? And so we see here that Sauvignon Blanc grape, they survived the longest, more than 60 days. Chardonnay grape, they survived more than 20 days. Everything else is more or less clustered around 10 days. The difference here, I wouldn't say that they do better on Sauvignon Blanc versus Chardonnay. These grapes were a year older, say that they were 3 years versus 2 years, and I think that's the difference there. And this is the other type of chart you're going to see a lot of today in my talk. This is a box plot. And they're the same data in a way, but without the trends over time. This is days survived across all the hosts. And this is 50 adults, mixed male and female, tracked until they were all dead. And here's how you interpret a box plot. So the top fence, bottom fence, those are maxes and mins. So maximum days survived, minimum days survived, and then you've got the 75% quartile, 25% quartile. And the line in the middle is the mean or the average, and the mean is what we use to determine statistical differences. And so you've got in bold above or below each box, that's the maximum, and then italicized, that's the mean. And then the letters, basically, if it has the same letter, those are similar, right? So A, there's only one A here because Sauvignon Blanc grape was so different from the others, okay? And so you see a wide variety in how the insects respond to these various host plants. And so next I'm going to talk about adult survival on citrus more, but let's look here at this. So the maximum survival was 13 days and the mean survival was 11.5 days. And so let's compare that original citrus experiment with our repeat of the citrus experiment. And first, let's talk about the methods. So our original test looked like this. We had our individual plants with bags over the foliage. We put 50 spotted lanternfly in each bag. The trees were about 5 feet tall, and we tracked them. A repeat looked more like this. The trees are bigger, bushier. We allowed them to put on fruit, and we put the whole plant in a cage so the insects could access the whole thing. Again, 50 insects, you can see one here. And the trees were about 5 feet tall, so this cage is about 5 feet tall, and they're kind of crammed in there. And the big difference is that the repeat, the citrus actually have fruit on them. We were concerned with fruit drop, and so we wanted to test that. Another difference is we found that the insects were a little more interested in mating than feeding, and that might be a problem. So what did the data look like? So here we have our survival curves and our box plots. So the original citrus is in yellow, the repeat is in orange, and they're pretty different, statistically significantly different. We weren't really expecting that, but we have some reasons why I think it happened. So the mean survival days for the original was 11.5, remember, and the mean survival for the repeat was only 4 days. And why is that different? Well, potential explanations. It's possible that the repeat, remember these trees had fruit on them, that fruit is going to take nutrients away from the plant. The insects don't probe the fruit, they probe the vegetative tissues of the plant, and those tissues might have been less good because all the nutrients, not all the nutrients, but a lot of the nutrients have been moved to the fruit. Another difference is the age at the start of the experiment. So the first run was done on August 15th, started on August 15th, and the second, the repeat started October 2nd. By October 2nd, the insects start getting a little more interested in mating than feeding, and so we think that might have contributed. We aren't sure how much each of these variables might have impacted it, but there are reasons why these data turned out different. Okay, another thing we learned from the citrus repeat is normally I pool the male and female data together, but in this case, I pulled it out and I looked at male trends and female trends. 25 of each, and we found that they are not statistically significant. So average survival for females, 4 days. Average survival for males, 3.5 days. That is not statistically significant. And so that's something we learned. We weren't really sure. We hadn't looked at them individually, but in this case, we found that males and females did not survive differently. Citrus. Okay, let's look at nymphal survival. Now we can also look at development. So see in this picture, we use the same setup for the nymphal work. The citrus trees are about 5 to 6 feet tall. The avocado trees, not shown here, were about 6 to 7 feet tall. Field collected first instars, added them to the trees, 50 nymphs per tree, 8 hosts for control plants. So we were dealing with 400 insects on each host type. And if the insects survive long enough, we also tracked their developmental milestones. So this is what the avocado and citrus data look like. We've got our survival rate over time, a box plot, and they're wildly different, right? Highly statistically significant. So for avocado, they died very quickly. So an average of 5.25 days, a maximum of 9 days. And for citrus, they did so well for so long. They survived and then all started to die kind of right at the end here. So maximum survival of 96 days and a mean survival of 93.38 days. Okay, and because they survived for all that time on citrus, we were able to look at their development. We didn't do that for avocado because, well, they died so quickly. And on nymphal survival, what we have here for this is a stacked box plot. Percent of each stage alive here, and then across the bottom, we have each of these bars represents an observation time point going out to the last one, which was 96 days. After that, they were all dead. So first instars, this is the dark blue here. They survived to day 19, and they started becoming second instars as quickly as 9 days. So 9 days to 44 days, we had second instars. 19 to 68 days, that's the dark purple. We had third instars, and we had fourth instars as early as 19 days. That's quite quick. But then we had fourth instars all the way out to 96 days. We had fourth instars for 77 total days, which is pretty surprising. And we had a single adult, which I had to highlight that because you might not have seen that little sort of pink bar, but one male adult emerged on day 68. So what are we going to say about this data? They survived for a long time. Yes, that's true. Did they survive well? That's a little more subjective. Normally, in the field, we don't have, it doesn't take them 96 days to move through their life cycle and become adults. When we rear them in the lab on hosts that we know they prefer, tree of heaven, grapevine, they develop much quicker. The fact that we still had fourth instars at 96 days is quite surprising, but they did survive for a long time. And the first time this has been shown, we did have a single adult emerge on citrus alone. Remember, they didn't have access to any other host. They just had these citrus trees. So SLF on citrus, what have we learned? So I've got a column of concerns and positive news. So concerns, they survived for a long time on citrus. Notice I didn't say well, because it should have taken them 96 days to develop through their nymphal life cycle. If they were on a host they do well on, for example. So we did find they can complete their life cycle, though, one male adult on day 68, but that's 1 of 400. So it wasn't a high success rate. They could develop to adult. We did find the nymphs produced a copious amount of sticky honeydew, and this has been shown to be a substrate for fungal pathogens like sooty mold. We did not allow this to exist in the lab. We wanted to hold the plants to see how the plants did, but we washed off all of the sticky honeydew. And in a orchard, that could be a much different situation, right? So in the positive news column, the adults did not survive for a long time on citrus, maximum of 13 days. Remember, the choice was not given. They were caged exclusively to citrus. We don't know what their choices would have been. Plants were not seriously damaged by spotted lanternfly feeding. They did not die. They were held for a while and they did well, even after 96 continuous days of nymphal feeding. And in the citrus repeat with the fruit-bearing trees, the fruit did not drop from spotted lanternfly feeding. However, the feeding didn't last very long, so we don't have definitive proof that they won't cause fruit drop. Okay, data summary. So the consensus so far is that grapevine is most at risk from adults. Lots of studies are showing this, but it's also important to remember that other hosts may support populations of adult SLF for short durations. This increases the risk of establishment and habitat where these hosts coexist, especially with grapevine. Nymphs can survive for a long time on citrus, 96 days, and can, in some instances, survive to adulthood on citrus alone. Nymphs produce copious amounts of honeydew while feeding citrus, and we worry that this could be a problem for areas where grapevine and citrus coexist. I'm thinking specifically of California. Broader implications, these pest-host interactions are incredibly complex in this highly prolificous insect. It's hard to know what they're going to do. For our purposes in a greenhouse setting, the responses to different hosts varies widely. And we find these differences can be dependent on life stage, and that's what you'd expect. In this example of citrus, adults only survived 13 days, but nymphs survived for 96 days. And more broadly, these interactions depend on things like host availability, host preference, which we didn't talk about, and a really important one, think about what the climate is in all the different regions, or what the weather is in all the different regions of the US right now. And it really depends on what hosts are available when each life stage needs them. If a nymph were to be in Pennsylvania today, they would get an ice storm. If they were in Atlanta, it'd probably look pretty nice. If they were in California, it would look wildly different. All of these different places have different climates. Okay, that's all the data I have. Acknowledgements, this is a team effort. Thank you for everyone who's contributed and to the funding agencies. And I'm happy to answer questions, and I will hang out in the chat. So if you don't get your question answered, I'll definitely get to it in the chat. Thank you.